Next part, we talk about the batch effect removal. Very often you have batches in expression. In fact, anytime you do a biological experiment with some replicate, you might end up having some batch effect. So these are non-biological variations. It makes the samples not directly comparable. Can you think of ways that batch effect happens? Yes. Yeah. In a microarray experiment, we usually only compare sample with the same array design. And imagine even with the same design, you bought the array this year versus you bought the array last year, maybe the company was making them differently. Or if you scan the array on 2 different scanners, or if these are the possibilities, you do the experiment on different days or months of the experiment, and it's the same culture dish, but the medium is slightly different. The postdoc were making up creating the media, maybe the serum is slightly different in this batch. It could be a different mice, say from a different company. You want to repeat the same experiment, but it's like from a different company. It happens, they will have different behavior. And yeah, different batches of arrays or scanners, different personnel, right? The person who is doing the array scanning might be different. In fact, in 2004, there are even studies showing that the atmospheric ozone level will influence the final array scanning or something like that. So yeah, definitely you have to remove this. Otherwise, you get totally overwhelmed with your batch effect. They look like a differentially expressed genes, but in reality, it's just a batch effect. So how do you spot them? So this is one experiment. You're growing some cell and you are knocking down one gene with 3 different, with different RNAi is basically an approach. You try to knock down the expression of some gene. And you do this in 3 different batches using different RNAi to do a knockdown of that gene. And so you can see the C's are the controls, and these are their batches: 2 in here, 3 in here, 1 in the top, right? The type is the treatment. R is the knockdown experiment, RNAi, and C is the control. And the third one is the different RNAi clones. So you can imagine we are using, say, 3 different RNAi. So RNAi 1 is like 1 point something. So RNAi 1.1 is using the first RNAi reagent, and it's the first to replicate, right? RNAi is also, the other one is using the first reagent on the 3rd batch. What would you expect in this experiment? If you do, so you do clustering, this is hierarchical clustering, and you look at this, what do we expect to see in this experiment? You have the original cell and you try to get rid of one gene. You try 3 different ways to get rid of this one gene, and you measure the gene expression, right? Don't you expect that the control condition to be clustered together and the 3, whatever the knockdown experiments are in the other condition? And maybe depending on the 3 different ways you try to get rid of this gene, you get those 3 kind of clustered. But in reality, what you see is really, the 3rd batch is all next to each other. The 1st batch is here, the 2nd batch is close to each other. And this is a clear case. So anytime you do clustering, the sample is clustered by batches, not by the biological conditions. That means you have a batch effect. And so you want to correct that. But question is, shouldn't the normalization take care of this? We've done, what did we do to do normalization before? Quantile normalization in RMA, right? Didn't we get rid of this? No, it doesn't. Yes, in fact, normalization and expression index procedures in the RMA only adjust the sample and probe effect. They don't really adjust this type of batch effect, and they have a huge effect. They are orthogonal to the batch effect. Yeah, the batch effect have big effect in your final experiment. Therefore, when you are designing an expression, whether it's microarray or later RNA-seq experiment, you want to be very, very careful. You want to record the day, the lab, the personnel, the environment, environmental variable. I think it's not as important, but at least you kind of keep track of which batch it is. When possible, you try to balance the group of interest. So supposedly, you can imagine if sometimes people have a reference sample every time they run a batch. They always correct everything according to that reference sample. But other cases, say you try to, in this case, look at lung cancer. As we mentioned, we collected the samples from different hospitals. They are now all in NCI and processed at the Broad Institute. So the Broad has two ways to process them. They say, this month we're going to process all the cancer. Next month we're going to process all the normal. Is that a good design? It's terrible, right? I think it would be better if they say every time we have the same patient, tumor and adjacent normals. Very often you take surgical samples, there is a piece of tumor, and then they take a nearby adjacent normal. You process those together so that you can compare at least the cancer normal from the same patient. And yeah, as I mentioned, we try to avoid the perfect confounding situation. When batch and group are perfectly correlated, for example, if you do your treatment in one batch and control in another, at the end, there's no way you can correct for that. It's all mixed together. You don't know whether it's a real treatment difference or a batch difference. Yeah, or treatment in one and treatment two in the other batch. So this situation is impossible to correct. Okay, it's good to have the treatment control in each batch. Maybe say your scanner can only scan 16 samples at a time. Every time you have 8 treatment and 8 control. Next time you have 8 treatment and 8 control, and so on. Okay. Yeah. So how do we really think about the batch effect? Oops, the samples. I'll correct that later. So you can imagine originally when you have a gene expression index, the columns are the samples and the rows are the genes, right? When we look at a linear model like Lima, if you watch the lecture video from last time, you can imagine we think the expression index of these genes in all these different samples are the following components. The first is the baseline expression. Oh, every gene is expressed at some basic noise level. And also we say, okay, supposedly these are treatment conditions and say 3 replicates of the treatment and 3 treatment of the control. This will be in you annotate your matrix. You say, okay, these are the same treatment condition. These are the 3 control condition. And because of this treatment control, they will have the treatment has an effect of this much on this gene, that much on another gene, this much on a third gene, right? So if it's a treatment sample, you add the effect of some treatment onto each gene. And of course, at the end, there's a noise, some random errors at the end, little things that you can correct. So these are just random variables. And in Affymetrix, in the Lima algorithm, what we're trying to estimate is this thing. What is that treatment effect, right? You're asking whether for that gene, whether that treatment effect is 0 or not 0 when you have treatment and control conditions. So now with similar approach, you can imagine batch effect is just another thing that you have to add to your original thing. Sorry, I need to add a plus sign in here. So originally you have your baseline gene expression. You also have your treatment and control, but on top of this, because the sample come in different batches, you can also put this in a design matrix to say, okay, these are your batches, batch 1, batch 2, or batch 3. And depending on the batch, they also have a differential effect on the different genes. And we don't know exactly what are the difference. I mean, we don't know why they create, but at least we know every sample with batch 1, somehow this gene is expressed higher. And so if we see that, we need to correct that. And of course, at the end, we also have the error. And so it's very intuitive, or intuitively similar to Lima's linear model. So you just consider batch effect as another kind of treatment, right? This is technician's problem. They're hand or whatever, they created a new effect on your gene expression, right? So in some sense, if you can estimate this, then you can subtract that out from your original gene expression matrix. Then you can estimate your real treatment and control difference. But if you don't correct for this, they will be muddled into this whole thing, and you will have much weaker ability to detect the differential expression just based on the treatment conditions. Okay? Yeah, so how do we detect batch effect? We use clustering. As we mentioned before, you can do k-means, you can do hierarchical clustering. You can see whether the different samples are clustered by the actual biological condition you want to compare, or they are separated by the batches. And if it's the second case, you know you have batch effect that you need to remove. And there are two different algorithms. The combat algorithm was, I think, still the most popularly used. With similar ideas, it's trying, you first have to define the batch, right? Okay, which conditions, if the batch is known, you say, okay, these are the first batch, that second batch, those are the third batch. And it will try to remove the batch effect from the original expression index. You end up with a new expression matrix with that kind of batch effect taken out from the original sample. And so you are left with the matrix that only has 1, 2, and the noise. Okay, that's it, that's the idea. And there is another algorithm called SVA. These are cases where you are not even aware of what type of batch there is, and it can automatically identify the batch and correct it. Most of the time, I think people do know the batch, and so especially if you keep track of your lab notebook, you will know to correct for batch effect using combat. How do you know whether your batch effect removal is successful at the end? Yeah, exactly. You cluster again, and hopefully after you run combat, the samples will be clustered by the biological conditions rather than the batches. Okay, that's batch effect. Questions? Questions?